The overall goal of this procedure is to isolate mutants in the parasite Toxoplasma gondii that are impaired for survival or replication following activation of infected macrophages in order to identify genes and pathways that are important for resistance to IFN-γ-Dependent cell autonomous immunity. This is accomplished by first empirically determining the dose of IFN-γ and LPS to be used in the forward genetic screen of the mutant parasites. The chosen dose should suppress replication of wild-type parasites following activation of infected macrophages, but still allow replication over a 30-hour period. The second step is to use the chosen dose of IFN-γ and LPS to activate macrophages in a 96-well plate that have been infected previously with mutant parasites and culture the cells for an additional 24 hours. Next, the infected cells are fixed, permeabilized, and stained with a polyclonal antiserum specific for Toxoplasma gondii and examined using immunofluorescence and phase microscopy to identify mutants that were unable to replicate or appeared in amorphous parasite-containing vacuoles following macrophage activation. The final step is to confirm the defect in each candidate mutant by evaluating its ability to replicate following macrophage activation in chamber slides that enable viewing of the parasite mutants at a higher magnification and resolution. Ultimately, immunofluorescence microscopy is used to show the mutant parasites replicate like wild-type parasites in unstimulated macrophages but are either unable to replicate or are amorphous following macrophage activation.
Though this method can be used to identify mechanisms used by Toxoplasma to evade the cell-autonomous immune response, it can also be applied to other systems by either changing the host cell that you're examining